welcome to the lecture starting the 6th unit here with meiosis. Before we get into actual meiosis though, we're going to do a quick review of mitosis. We talked about mitosis in Unit 4 and I want to do a quick review so that we can apply those concepts to this new meiosis concept. So first, you should remember that the function of mitosis is the division of those somatic cells, or those body cells, that results in two genetically identical daughter cells. We know they need to divide for growth, repair, and then asexual reproduction for those unicellular organisms. You should remember the phases: interphase, prophase, metaphase, anaphase, and telophase. These are going to come back in meiosis, so please make sure that you review those if you do not remember them as those stages of mitosis. Once again, just to make sure to go through those again, make sure you know what goes through each of those phases. And then once again, that end result of mitosis is two identical cells with the same number of chromosomes as the parent. So you're ending up with identical cells in the end as you did with the beginning. So now that we reviewed mitosis, now we're going to look at types of cells. So first, there's two different types of cells, haploid and diploid. Haploid cells, we use an N here to show that it has half the number of chromosomes in the cells. So half compared to the original amount of chromosomes for the organism. While diploid is 2N, which is a full number of chromosomes for the cells. And so our somatic cells are diploid, or 2N. They have a full set of chromosomes. Our body cells do. But our gametes, you'll notice, has half of the number of chromosomes, and we're going to talk about why later. But you need to know that the gametes of the sex cells are haploid and they have half of the chromosomes as a normal somatic cell. So once again, somatic cells are body cells that are diploid, and then our gametes are sex cells like sperm and egg, and they're known as haploid, or have half of the DNA. Do a quick review of chromosomes real quick. Chromosomes are made up of DNA, and a segment of DNA that codes for the making of a protein is called a gene. So you have genes on those chromosomes that make proteins, and you know those proteins, when they're made, then they're expressed. Well, how many do we have then? In our somatic cells, we have 46 chromosomes because they're diploid, and so you have both sets of chromosomes in there. While our gamete cells only have 23 chromosomes. They're haploid. And so once again, our body cells will have 46, and our sex cells are going to have half. They're haploid with 23. So how does that relate to meiosis? Meiosis is the production of gametes, or sex cells. And so if we know that gametes are haploid and they end up with half of the DNA as a normal somatic cell, then we need to know the amount of chromosomes that are in those cells. So once again, meiosis is the production of gametes. The number of chromosomes in the original cell is going to be reduced by half for the gametes. And then the cells that go through meiosis are sex cells, and it only occurs in your sex organs. So whether that's a male creating sperm in the testes or a female creating eggs in their ovaries, that's the only place where meiosis is going to occur, because it's only reproducing sex cells. So again, males have the sperm and females have the eggs. What is the end result of meiosis? Meiosis produces 4 genetically different haploid gametes. And so this is the opposite of what mitosis does, right? Mitosis only creates 2, and they're going to be genetically identical with a full set of chromosomes. So they would be considered diploid. But meiosis will have 4 genetically different cells in the end, and they're also going to be haploid, which means they have half of the DNA as the original. So if you see here, we have 4 chromosomes in the beginning, but at the end we end up with half of the DNA in each of those cells with only 2 chromosomes remaining in each of those. So they would be considered haploid. Now the reason that they're haploid is because once you fertilize, you're going to mix the sperm and the egg, they're going to come together, and you're going to end up with a full set of DNA in the end, because you get half of your DNA from your mom and half of your DNA from your dad. And that's why we say you're 50% made up of your mom and 50% made up by your dad. So let's overview meiosis. Let's see what it looks like. So first we have our parent cells here, which have a full set of DNA. That full set of DNA is going to be duplicated, just like before mitosis. It needs to duplicate. So it's going to duplicate, then you're going to have what we call homologous chromosomes, or chromosomes that are homologous to one another, identical. Then those homologous chromosomes are going to separate into two separate cells after the first stage of meiosis, after meiosis 1. And then those second set of cells are going to go through the process of meiosis again, meiosis 2, but they're not going to replicate before, and that's how you end up with 4 cells that have half of the DNA as the parent cell. So let's look at the 2 phases. We have meiosis 1, and that's where the homologous chromosomes are going to separate into 2 diploid cells. So they're still diploid at that point. And then meiosis 2 is going to take those chromosomes, separate them apart, and form 4 haploid cells. So if you remember, you see from here, you end up with 2 diploids, still have 2 chromatids here in each of these, and then you end up with 4 haploid, or half the DNA in those. So we know that it goes through 2 divisions. Meiosis 1, once again, separating the chromosomes, and then meiosis 2, separating the chromatids. Once again, 4 haploid cells with half of the DNA at the end. So, before meiosis 1, the chromosomes need to replicate, the DNA needs to replicate, just like before mitosis. It needs to go through a phase of replication in interphase. So they're still going to have that S phase before interphase. And then once it's ready to go into meiosis, it starts with prophase 1. Now we put a 1 after the prophase to indicate it's the first time going through prophase in meiosis. If you remember from before, meiosis is going to go through 2 sets of division, and so we're going to identify which set of prophase that it's going through because it's the first time it's going through. And just like in mitosis, the chromosomes are going to condense, so you're going to start to see the chromosomes form. You're going to see homologous chromosomes starting to pair up. So the ones that are paired with one another, you end up pairing them up. And then you start to see crossing over occurring. So what are homologous chromosomes? Homologous chromosomes are chromosomes that occur in pair that are similar to one another. So that means they code for the same traits. So these 2 are going to be homologous chromosomes from 2 different parents here, right? They're going to have different traits because one parent is going to have a different eye color, for example, or a different tongue type, or a different hairline. But because they contain the same traits, we call them homologous, because this chromosome for this parent and this chromosome for this parent, the gene here is for earlobes, whether you can roll your tongue or not, your hairline, and your thumb. And so since they have the same genes, the genes might not be exactly the same in specifics, but the types of genes, the traits, they're considered homologous because they can pair up because they are coding for the same traits. You're going to have 23 sets of homologous chromosomes between your mom and your dad. You get 1 set from your mom and 1 set from your dad. But also in this prophase, we talked about crossing over, right? The chromosomes condense, those homologous chromosomes paired up, and then crossing over occurs. What's crossing over? Crossing over is the chance for the chromosomes to then exchange genes. So the homologous chromosomes are going to separate during meiosis, but they end up coming together before that, and can easily cross over, like you can see here, where one leg of one chromosome can end up switching with a leg of the other and end up switching some material from one to the other. So it's just one way to increase genetic variation during that time. When they pair up together, they can end up exchanging some of their genes, so then when they end up giving to their offspring, it can increase that genetic variation. So this is also a reason why in the end you're going to end up with completely different DNA in the end result of the meiosis, because this genetic variation can also occur. So you can see here, these would be the original chromatids, but now these are the end resulting chromatids after crossing over, because they've increased that genetic variation, they've changed it up. So after prophase, you still have metaphase. If you remember PMAT, it's still in the same order, but different things happen. So in metaphase 1, the first time it goes through metaphase in meiosis, The homologous chromosomes are going to move to the middle and attach to the spindle fibers. So they're going to still go together in their homologous chromosomes. They're still paired up, and they're going to meet in the middle of the cell, just like in regular metaphase. Then in anaphase, the homologous chromosomes are going to start to separate. So you can see the full chromosomes physically separating from either side of the cell. Now, this is also an area where non-disjunction can occur, which is the unequal splitting of chromosomes. And so there are times where maybe a spindle fiber here can attach to this chromosome and end up pulling it over here to where you end up getting 3 in one cell and 1 in the other. And that is called non-disjunction, when they don't equally split in half. Non-disjunction can occur at 2 different points. It can occur during the first division, during anaphase, or it can occur during the second division in anaphase. So you see here, non-disjunction occurred here, and now you ended up with 4 chromosomes in this cell, but none in this. And then when it split in the second phase of meiosis, then you ended up with 2 in each and none here, instead of 1 in each, which is what it should be. Then here's the example for it happening in the second phase. You ended up dividing properly here, but here you ended up with non-disjunction, so you ended up with too much genetic material and not enough here. Non-disjunction can lead to certain chromosomal disorders like trisomy 21, where you end up with 3 21st chromosomes, and that's the disorder for Down syndrome. And so that is how things like Down syndrome can occur. Next, telophase 1. So after that anaphase, you end up with telophase. The cytoplasm begins to divide, and you start to see the 2 nuclei coming back. And at this point, the 2 new cells still have the same number of chromosomes as the original cell, because if you remember, before meiosis ever began, it ended up replicating that DNA. So you had twice as much DNA, and then it's just splitting it back down into its normal set of DNA. But then the cells are going to go into meiosis 2, which is just like mitosis, like before, but it doesn't replicate in between. It goes straight after telophase and cytokinesis. It goes straight back into meiosis 2. It's not going to have a chance to replicate those chromosomes, which is why in the end we have half the DNA. So in prophase 2, the chromosomes are going to still condense, and the nuclear envelope is going to break down again. Metaphase 2, they line up in the middle. Anaphase 2, you're going to end up separating those sister chromatids to opposite sides of the cell. Once again, non-disjunction can happen here too. And then telophase 2 is where the cytoplasm starts to divide, and then you end up with 4 haploid cells with half of the DNA as the original cell. So after meiosis, you produce your gametes, and once those gametes are produced, then it allows for sexual reproduction to occur. So sexual reproduction is when 2 haploid cells, a sperm and an egg, makes 1 cell. That's a diploid zygote. So once this sperm penetrates this egg over here, they're going to share that DNA. Half the DNA is in the sperm, half is in the egg, and then you end up with a diploid zygote. So it's no longer haploid because you take those 2 halves and make a diploid. This process is called fertilization. And then after this has been fertilized, now mitosis can occur, where the cell can end up dividing to create identical cells, just like your body cells, to create a new organism. So once again, the egg has a haploid gamete, right? The sperm is a haploid gamete. You have half of the chromosomes in each of these. They come together and fuse to create the diploid zygote, which has the full set of chromosomes, and then those divide through mitosis to create you. Okay. Once again, fertilization is the process in which the egg and sperm unite. A zygote is a fertilized egg, and an embryo is an organism in the early stage of development. So just a reminder, meiosis is the division of these gametes, these sex cells. You're ending up with 4 genetically different haploid cells with half of the DNA as the normal cell. And then mitosis is the division of body cells, where you end up with identical cells in the end. Okay. Now you're going to complete the comparison and contrast activity between mitosis and meiosis while watching the mitosis versus meiosis video for the Meiosis sisters. So this is what's going to be due at the end of class today.